very schematically. So you have an animal, maybe a rat or a mouse, to treat the animal often for prolonged periods of time, up to lifetime. And then, in principle, what you observe is the animal is dead or healthy, or it's diseased, or it has cancer, or something like that. And then the veterinary pathology comes into play. Up to 50 organs are analyzed for abnormalities, and histology is performed. And that's more or less it. So it's described where, in which organ toxicity occurs at a high dose. And it's not questioned what the mechanism is that is leading to that kind of toxicity. That's classical toxicology. It's still the way it is undertaken in chemical industry, pharmaceutical industry, not in cosmetic industry because there it's forbidden to use experimental animals. But according to guidelines, these studies are performed. The point is, mechanistic aspects are not really regarded here. And this changes. Toxicology really changes, and I brought here the conceptual framework that was developed in CERAT 1, which is a very large project sponsored by Cosmetics Europe. Maybe our next speaker will briefly talk about that. And the European Commission, it's a large project where, as you may know, ToxBank is one of the partners. And ToxBank is coordinated by Mary here in our chair today. I have the pleasure to be partner in COACH, which is a coordinating action, and together with Maurice, we are coachers. We are trying to somehow bring this large project running, keep this running. Now, I don't want to really guide you through this conceptual framework. I just want to make some points. Very different from a classical approach. First of all, we do not use animals, of course, but we start with hypothesis generation. So we ask the question, how might this chemical that we are investigating produce toxicity? What is the mechanism? And here we have to discriminate between the type of adversity. That will be the next step. So what kind of adversity do we have? Do we have a general adversity which will affect many different organs and many different tissues? Or do we have an organ-specific toxicity? And you have to know that very often the discrimination between these two types of toxicities are due to admin properties. So toxicokinetics will determine whether toxicant is toxic in a certain organ. Take a very simple example. When you have, for example, a toxicant that is toxified in liver cells, then it will be toxic in the liver. So toxicokinetics are to be taken into account. And then you come to the part of toxicodynamics, and here again you have either many biological targets. Take an alkylating agent, for example, will alkylate everything in the cell, will be toxic in any cell. But you can also have a target that is present in almost any cell. We have heard about the H receptor. The H receptor, for example, is present in almost any cell in our body. So here we have a specific target, but the target is present in almost any cell. And that's different, of course, for the target organ, where, or for organ-specific adversity, where a specific target is hit if it's for toxicodynamic reasons, because, for example, the target is only present in a certain cell within our body. Now, how can we discriminate between these two types of adversities? We can do that, for example, by using information from different sources. In this case, what I would like to show you very briefly is our results from the ToxCast program run by the US EPA, where approximately 700 high-throughput assays are combined, covering something like 300 different signaling pathways. And there's an interesting study that was published by Rusty Thomas and colleagues in 2013, where he has investigated the Toxicology data set and asked how many of these assays that were present in the Toxicology program were activated by a specific compound. And he said a line of discrimination at 10 assays. And what you see in this graph behind me is that about 80% of the chemicals hit 10 or more of the assays. Only 20% were specific. And according to this definition. And why is that important? Because if you have a chemical which is unspecific in its mode of action, then you do not need a specific assay. So you do not need to screen with many specific assays. You simply, if you know this particular mode of action, a very small number of assays will be sufficient. In particular, if you have some information on the mechanism of action of this chemical for testing that. Now, another example which I would like to give you comes from the project that I was coordinating 5 years ago. That project ended, called ReproTect. And at the end of the ReproTect project, we conducted a feasibility study, we called it so. And in this study, you see, with abbreviations, you see, I think, 20 different assays covering the reproductive cycle at different points, so very, very different endpoints which are measured here. And we run 10 test chemicals through these 20 different assays. And what you see here, I have to explain that first, is you see the results with 3 compounds, 4, 2, and 5. And at the x-axis, hard to read, that's a scale of concentration of the assays, of the different assays, where the chemicals were run in the different assays. And each of the points that you see here is a result obtained with reference compounds, except where these points are further indicated with these here, like this here. When you look at this, what you immediately see is compound 4, there was no activity. It was a reference compound without any activity. Compound 2 here has a very specific effect, has a very low concentration range. These are unspecific effects here. But this compound here was positive in almost any assay. So if you see that, then you immediately know a couple of things. But among these, you know that compound 2 has a very specific effect, while compound 4 is an unspecific toxicant. In fact, compound 4 was cadmium, so that will affect almost everything in the cell, and compound 2 was an inhibitor of a specific enzyme with highly specific activity. You can do more with that, and with this kind of data. And I'd like to show you that with this graph here. Again, you see the concentration range here, you see the reference compounds, and you see, for example, compound 3, one of the 10 test compounds, was located at this concentration range. And what you can do now is you can ask what is the nature of the nearest neighbors to that compound, concentration-wise. And here you can do that with one assay, but you can do that with in this case 5 or all 20 assays. So you have the information on the activity of your test compound relative to the reference compounds, and you know the mode of action of the reference compounds because you have chosen them as reference compound in your assay because you know a lot about this. So you get some kind of signature, and it was amazing that this type of nearest neighbor analysis, done by hand, by the way, not automatically, that this, what kind of information that delivered. So, to sum that up, integration of data from different sources will tell you something very important about mechanism of action of your test compound, and it may, of course, predict the increase the predictive power in your assay. But you need to have suitable tools for integration, and with that, I come to a program which I would like to, well, Barry and his group knows that already. It's called InCroMAP. I would like to make it noticeable to those of you who don't know it, because it's a powerful tool which is suitable for integration of data coming from different sources. It was developed, in the group of Andreas Sell, a colleague of mine in Tübingen, within a project which is called MARCA. MARCA is an IMI project where both Andreas together with his colleagues were partners. So what is InCroMAP? InCroMAP is a tool useful for integration of data. What you see behind me here is, in principle, a schematic outline of the MAP kinase signaling pathway. And you see changes indicated in various parameters, in this case, HRAS mutated mouse liver tumors, which we have analyzed here. And what you can do with this tool is integrate data on messenger RNA, protein, phosphoprotein, DNA methylation, and metabolome if you have this. Metabolome data are not in this graph, but the tool is suited to do that too. So what can you do? I exemplify here this kinase where you see in red an increase in the expression of the messenger RNA. The protein, the basic protein, is unchanged here, but there are 3 phosphoproteins which in this case are increased in level in these tumors. Here in front of the genes you see a box which indicates promoter methylation. If you have promoter methylation data, you can also visualize these data. If this bar is very narrow, it's little methylation. If it's broader, small methylation promoter. If it's going to the left, it's hyper. If it's going to the right, it's hyper. If it's going to the left, it's hypomethylation. So you immediately see for these genes the promoter methylation present. And you can ask different questions, by the way. You can look for global methylation, you can look for specific methylation, and things like that. And you have information here also on microRNAs which were measured in this experiment, and their targets, either predicted targets or validated targets. And you can immediately compare the changes in microRNA expression levels with changes in genes. You can do more. You can overlay your experimental data with, in this case, for example, the KEG pathway. Now, what you see here may look to you like an underground map or something like this. To me, it's very telling. I'm a wet lab experimental person, and I know, for example, that this is TCA cycle, this is lipid metabolism, this is glycolysis, sugar metabolism, this is drug metabolism, and so forth. So I can immediately orientate myself in such a map. And what you see is there are changes here in 2 tumors mutated in 2 different genes. And when you compare these two, what you immediately see is that in the rest mutated tumors you have changes in this case messenger RNA expression for genes relevant for lipid metabolism, which you do not have here. And it fits nicely with what you observe because the rest mutated tumors are lipid storage tumors in this case. So for me as an experimental person, this kind of visualization is very helpful, and InCroMAP is a tool which I would really like to advertise because it's very helpful in this respect. Now what is important, we've heard that at Moni already, if we have a bioinformatician that will generate such a nice program, and if you have a wet lab researcher like myself, first of all, they need to speak a common language. I need at least to partly understand what this guy is doing, and I have to develop this program together with him. So we have to interact. We need to have a common language, and we need to have a common ontology. And that's a problem because in toxicology, we do not really have a common ontology yet. And definitely, I was talking to Barry about that. OpenTox could be a program where a common ontology could be assisted in development. Now, I'll like to give you another example where you see that the interaction between the wet lab researchers and a modeler in this case is very important. And this is this series of, I think it's 2 or 3 slides, is a gift from Jan Hensler, and it represents work that was predominantly done in his lab. And all these people here mostly are from his lab, wet lab, experimental people, but Dirk Rasto is from France. He's a modeler. And the question here in this study was which mechanisms control the reorganization of functional liver tissue following chemical insult. So the model they have is carbon tetrachloride. If you give that to an experimental animal, what you see is that in the central lobular area of the liver lobules, you kill all cells. All cells are eliminated at day 2, but at day 8, what you see is there is complete recovery. And the question was, how is that organized, spatially and with respect to timing? And the model they came up with initially was this one here. You see the damage initially, and then you see these brown colored cells, and simply the assumption was they randomly divide, fill up the gap, and that's it. Unfortunately, this model did not fit with the data that were observed. It was simply wrong. And to make a long story short, what turned out to be correct, first predicted and then shown experimentally, was that there is an interaction between the endothelial cells of the sinusoids in liver and the hepatocytes, and the hepatocytes divide and orientate their spindle after an initial orientation, aligning to the endothelial cells, and that model then fitted well. And later, 2014, there was a paper appearing there where we have the molecular explanation of that interaction between the 2 cells. So there are signaling molecules which are exchanged between the liver sinusoidal endothelial cells and the hepatocytes, and the signals are now better understood molecularly. So what that tells you is you need interaction when you want to be successful in what I would like to call modern toxicology. You need the interaction between particles from different disciplines. Biologists, bioinformaticians, modelers. The toxicologists you still need because somebody needs to know about the chemicals that you administer, and sometimes it's also helpful in model generation. But these people have to work together, and when they work together in a good way, that's trivial, but it's important. Together they are strong. Together they are better as if they work alone. In principle, I'm at the end, but what I would like to advertise here, I'm a SURAT1 person. I'm a coach in SURAT1. We have a final meeting of SURAT1 at the 4th of December in Brussels. Unfortunately, you cannot read the program, but it's very easy because go to the homepage. If you like, register immediately or read the program first and then register, and come to the meeting, because I'm very sure it will be a very interesting meeting where you will see the scientific results of what has been produced within SURAT. You will learn a lot about toxicology because we have a kind of a training course there, an educational course where we have a case study where you will be guided through our case study, and you will see how these technologies work together to ultimately come to answer the question, is that dose safe or is it not safe? I have 3 of these kind of flyers brought with me. Unfortunately, I didn't have more. I have 3 here, and I have a flyer of SURAT here, which you can also take if you wish. And with this, I thank you.